Hi guys, so the paper that I looked at is titled Specialized Coding of Sensory, Motor, and Cognitive Variables in the VTA Dopamine Neurons. It was written by Englehard et al. In 2019, and basically what these researchers wanted to find out is whether or not the dopamine neurons in the ventral tegmental area had the same function or multiple functions. So the ventral tegmental area, it's right there. It's a portion of the midbrain that is highly involved in development and expression of behaviors associated with drug use. So there are a lot of dopamine neurons in the VTA. We know from past research that dopamine neurons are highly involved in reward processing. So whenever a behavior is rewarded, dopamine neurons are activated. So in order to study these dopamine neurons, the researchers took a bunch of mice, put them in a virtual reality maze situation, kind of like that one, where the mice thought they were in a maze, and they would be cued by the researchers to turn a certain direction. If they turned the right direction, they were given a reward of water, and if they turned the wrong direction, there was a tone and the mouse was given a timeout. So the researchers found, shown in this graph, that during the cue period, which was when the mouse was cued to go a certain direction, the dopamine neurons had a lot of different functions. There were a lot of subsets. But during the outcome period, which is when the mouse was either rewarded or punished, the dopamine neurons all basically were involved in reward processing. So in order to actually image these neurons and find out what was firing when, the researchers injected a drug called GCAMP into the mouse's brain above the VTA, and this drug glows whenever there is neuronal activity. So using this drug and a 2-photon microscope, which is basically just a very, very strong microscope that allows the researchers to see beneath the surface level of the brain at the neuron level, using these technologies, the researchers were able to actually see the change in fluorescence of a specific neuron and find out, when it was activated and what behavior that corresponded to. So here there are 8 graphs of change in fluorescence of these dopamine neurons. Each graph corresponds to a different variable. So for example, the first one is position. It's blown up a little bit over here. So the researchers found that 91 out of the 303 dopamine neurons imaged was involved in position, which basically just refers to the mouse's position in the maze. So each line in this graph of all these colors corresponds to a neuron, and the color basically just means the neuron's change in fluorescence, so how much it fired over the course of the maze. To make it a little easier to see, the researchers took out one individual neuron and made a linear graph up there, and found that as the mouse progressed through the maze, as its position increased, the firing rate of this dopamine neuron also increased. So this is just an example of the many functions that these dopamine neurons actually have and the factors that go into their firing rate. So that is all I have for you guys today. Thank you for watching.